Hello, this is Mrs. Ness-Deden, and today we're going to talk about protein structure and protein folding. This is something we've mentioned already a couple times this week. The fact that changes in DNA can sometimes change the shape of a protein, but today we're going to look a little more in detail at how that happens. So we're going to start by making some observations on an interactive here. So here you see a sequence, a strand of DNA. And this interactive, you can actually watch transcription and translation. We're going to jump right to the end and take a look at the protein that would be made based on this DNA sequence. So this is an animation of the protein. All molecules are constantly moving. That's why it's kind of vibrating. But what I want you to do right now is make some observations of the shape of this protein. What do you notice about the basic shape of this protein once it's folded up like this? Now we're going to make some changes. We can go in here and edit this DNA sequence. So let's start just with a substitution mutation. What if I change this G to an A? Let's see if that affects the shape of our protein. So as we talked about, substitution mutations sometimes have a big effect, sometimes they have a small effect, and sometimes they don't have any effect at all. What if we go in and make another mutation? What if we, this time, delete two bases out of here? So I'm going to take out these two T's in the middle completely. So that's a deletion. We removed two bases. That made a bigger difference in the shape of our protein, right? We have a couple more green amino acids, so some of them have changed what they were. What if we go in here and insert a section of DNA? What if we insert a GCCGG? Now what happened? So you already know that the sequence of DNA affects the shape of the protein. And like I said, today we're going to talk a little more specifically about how and why that happens. So when your cells read the DNA, they transcribe it into messenger RNA and then translate it into a sequence of amino acids. That's what this picture right here is. Each of these little circles is a different amino acid, but this chain of amino acids that's formed is referred to as the primary structure of a protein. It's not a protein that can go out and do its job yet, but it's kind of the first step in building a protein. The amino acids are put together in a particular order, and as you've been learning over the past couple weeks, that order is encoded in the DNA. That's what those DNA instructions say, is the order of amino acids. There are 20 different amino acids that exist in nature, and so all polypeptides, all proteins, are made up of some combination of those 20 amino acids in a specific order. 12 of those amino acids are made by your body. You manufacture your own amino acids. The other 8 you get from your diet. And so we refer to those as the essential amino acids. We actually need to have all 20, but those 8, it's essential to get in your diet so that your body has all the building blocks it needs to make those proteins. Each of these proteins has its own unique chemical properties. You can see on the right here, this is a diagram of the chemical structure of an amino acid. This top part is actually exactly the same in every single amino acid. And this is the part that kind of links together to form the chain. But this part, what's referred to as the R group, is unique and different on every single amino acid. It's a bunch more atoms right there that have their own chemical properties. This chart right here shows you more of the chemical structures. And this isn't something that you need to know or memorize, but it's important to realize that each amino acid has that R group. That's the colored section here. And so here you can see how very different those R groups can be. This diagram has them kind of grouped according to some of their chemical properties. This top part, all of these green amino acids up here, the R group on those is hydrophobic. It's nonpolar, which means it doesn't want to mix very well with water. The second group with the pink R groups here, these are hydrophilic. They have a polar side chain, and so they do dissolve well and want to be near water. And then at the bottom, there are some R groups that have a negative charge and some that have a positive charge. And all those different chemical attributes affect how those amino acids are going to interact with each other and how that chain is going to start folding up on itself. So we're going to look at another interactive here to kind of see how that works. This one focuses again on those that characteristic of being hydrophobic and hydrophilic. In this case, the green amino acids are hydrophilic and the yellow ones are hydrophobic. So if you keep that in mind, take a look at this and predict how you think this is going to fold up. If the yellow amino acids are hydrophobic, where are they going to try and go? And the green ones are hydrophilic, so where are they going to try and go? Keep in mind, living things are mainly composed of water, so all of this area around the protein is made up of water. So let's see if you're right. We're going to watch this protein fold up just based on whether those amino acids are hydrophobic or hydrophilic. So you can see here, most of the hydrophobic amino acids, those yellow ones, tried to kind of move in towards the middle. They don't want to be out here by the water. They want to be in the middle away from the water. So they moved into the middle as much as they could. Whereas the hydrophilic ones, the green ones, are out towards the edge because they are attracted to water. They want to be out there. How do you think this protein shape would change? Look at the shape of this protein that we made. How do you think this shape would change if most of the proteins were hydrophobic? We're going to try it here. So if we change this so they're mostly hydrophobic, look what happens. Obviously, not all of our amino acids can get all the way to the middle, but you end up with a protein that's a much more kind of blobby, globular shape. What would you predict will happen if we change them so they're mostly hydrophilic? So here's what happens if we do that. If we have mostly hydrophilic proteins, you can see the shape of this protein changes quite dramatically. Now, we've been changing all of the amino acids or most of them each time we do this. Even if we just change one, though, if we change this very left amino acid at the very left end of that, if we change that to a hydrophobic amino acid, it wouldn't want to hang way out here anymore. It would come all the way back into the middle, and that would completely change the shape of this protein. So hopefully that helped you see a little bit how those interactions between the amino acids can affect the shape of the protein, and how even if we change one, that can change the shape of the whole thing. Hydrophobic and hydrophilic interactions are just one characteristic of those R groups of the amino acids that causes a protein to fold, though. So this picture right here represents a chain of amino acids. That's what this kind of orangish band is. And they gave you some examples of the R group. So this amino acid kind of right here has a hydrophobic R group. So that hydrophobic R group is trying to hide in the middle. Same with this one right here. They're trying to kind of hide in the middle. So that's pulling the protein together there. Here we have an example of an R group that has a positive charge. So that's attached to that amino acid. And this one over here has a negative charge in this part. And if you remember anything about positive and negative charges, opposites attract. So these two are attracted to each other and form a bond that kind of starts to pull this part of the amino acid chain and this part of the amino acid chain together. Same thing over here. These R groups form a hydrogen bond. And so you've got this part and this part that are kind of attracted towards each other, and they start pulling the protein together here. So they're all types of different chemical interactions that are happening that are pulling this amino acid chain into that three-dimensional shape. And it has to do with the specific chemistry of those amino acids. And so if we change the amino acid at one particular point, and the R group has a different chemistry, that's going to change these interactions that are happening and change the three-dimensional shape that we end up with. So to kind of put this all together, this goes through the whole formation of a protein. We start with that chain of amino acids, and as soon as we put those into their specific sequence, they start to pull and fold each other up because of all those chemical characteristics of the amino acids themselves into a unique three-dimensional shape. And remember, when it comes to proteins, the shape is what determines how it functions. So if the shape changes, the function's going to change as well. So we've been talking this week about mutations in DNA. And so to kind of tie this all back together, if there's a change or a mutation in the DNA, that can lead to a possible change in the amino acid. Remember, sometimes if the DNA changes just one letter, it could code for the same amino acid, in which case there's no change and there's going to be no effect. But it might change the amino acid. And if the amino acid is different, that will change the shape of the entire protein, either a little bit or a lot, depending on the chemistry of the amino acid and how it changed. And if the protein's shape changes, that's going to change its ability to function. It might not be able to do its job in the living thing at all. It might not even be built completely. In very rare cases, it can actually make it function better, but much more often, proteins that have changed their shape can't perform their function and don't work well. And that can be the cause of some different genetic conditions and genetic diseases. We'll continue to talk about those a little bit more as we continue through this unit. Have a great rest of your day. That's where we're going to leave off for today. Check Student View, contact your teachers if you have any questions, read the post on Google Classroom to make sure you've done everything for today, and have a great rest of the day.